Hello, everyone. I'm Neil Druve, a health program specialist at the National Institute of Neurological Disorders and Stroke in the Office of Neural Exposome and Toxicology. Thank you for joining us today for this mini workshop on the right tools and resources for neural exposome research. So we've had many people register for this workshop, more than 175. So we're really thrilled that you were able to join us. I'd like to mention a couple of housekeeping items as we get started. We've set this up as a Zoom webinar, so you won't be able to unmute. If you have a question during one of the presentations, we encourage that you enter it in the Q&A box. We're monitoring the Q&A box and the chat, and we'll be able to direct your questions to the speakers and the panelists. You can also activate captions under live transcript at the bottom of your screen. And we are recording today's workshop. Afterwards, we'll make the we'll make the recording viewable to everyone, and I'll send all the registrants a link once that is ready. Next slide, please. Okay, so just to start this workshop, we were going to have Dr. Pam Line, who's the chair of the Strategic Planning Committee for the Neural Exposome, but she's unfortunately unable to make it, so I'll give a brief overview of the strategic planning process. Next slide. And okay, thank you. So thank you for joining us for this workshop. It's part of a larger strategic planning initiative that we have at NINDS to identify the priorities for NINDS in the area of the neural exposome. So our committee has been hard at work. They've been meeting since the fall of 2023. We've had a series of meetings throughout, through the winter, through the spring. And these will culminate with a final meeting in mid-July. That is going to be an open meeting, so we will advertise about that. I hope you're able to join us. And as part of these discussions where our committee has been, defining the exposome research within the NINDS mission, prioritizing areas of neural exposome research, identifying best practices for communicating and engaging with neuroscientists about this exposome research. We've also been interested in kind of further development on two fronts, and we've had many workshops to follow up on these. One of them is to discuss and identify the best practices for fostering collaboration between NINDS, other NIH institutes and centers, and building teams to perform neural exposome research. So we had a mini workshop on team science at the end of February. And another charge that we've had, that we've given to the planning committee, is to identify tools and resources that will advance neural exposome research. And that's what today's workshop is about. So finally, after the last meeting of the planning committee, that's in July, they'll form their report to the NINDS Advisory Council. That will be due in September 2024, and then NINDS will present an implementation plan to Council in February 2025. So that gives an overview of where we are, and to move us on to the remainder of this workshop, I'd like to introduce Dr. David A. Jett, Director of the Office of Neural Exposome and Toxicology at NINDS. Dr. Jett's scientific interest is in the impact of chemical agents on nervous system function, including the molecular and cellular mechanisms of cognitive and neural development. Dr. Jett has served on White House and intergovernmental committees that set the nation's research priorities, as well as science advisory panels for the Environmental Protection Agency and the Department of Defense. Dr. Jett?
All right. You hear me okay?
Yes.
All right. Well, hey, everybody. And thanks, Neil, for the introduction. So this will be a short presentation on what we're doing in exposome research at NINDS and why tools and resources are so important if we're really going to get anywhere. Uh, so as you know, the focus of the NINDS is on the brain and the nervous system, and we're more of, I guess, a disease-driven institute, but with a strong basic research component to figure out the causes of these diseases. So I guess the first thing is, what is the exposome? It is not the exosome. I'd like to say that this is the next step after the human genome project. And so we know genetics have done a lot in terms of insight into the etiologies of inherited diseases, but the fact is that most health risk factors cannot be explained by genetics alone. And so the word exposome that was coined by Dr. Wilden back in 2005 is really all the environmental exposures affecting the genome. Now it's really all non-inheritable factors affecting gene expression and across the lifespan. So this is a new frontier. I think this is going to unlock a more holistic approach to disease prevention and more effective personalized interventions. So yeah, the exposome is not really new. As you can see, all these numbers of papers that have been, and they're just still growing. And there's tons of different ways of looking at the exposome that you can see. And one thing that I see, that it seems like all the exposome talks that I see are now using this movie advertisement, because really in its broadest sense, you can make the argument that it's everything, everywhere, and all at once. So again, we were talking, many of the neurological disorders have these complex etiologies, and they include these non-inheritable factors, and we're calling them, for us, the neural exposome. And as this effort was growing, we put together a paper in one of our journals, major journals, to describe our initial thinking for the office. And it includes our own view of what the neural exposome looks like. And I know there have been many of these figures, and not two of them are identical, but this is our first sketch, I guess, for us. And as you can see, there's three areas that we think are good for really talking about the exposome. One is the exogenous factors, like things like, of course, environmental toxicants, but also things like climate change and light and noise. Also endogenous factors, including genetics, but also things like the microbiome. And then there's behavioral factors, psychosocial, of course, but again, also things like lifestyle and sleep and stress. And one thing to remember is that a true exposomic research project would include sleep, as well as, say, air pollution, as well as microbiome, et cetera. So as you can see, things like team science and the right tools and resources are really important for this kind of research. So we do have an early focus area that really will require these tools and resources. And I say that they're early focus because, as you've heard earlier, that we have a group of experts working on a strategic plan that will help us decide what our final priorities will look like. And as you can see, they're endogenous, exogenous, and behavioral factors. And we've done some work on a lot of these already, things like a microbiome and others. So these are some of the reasons the exosome, I think, is timely, with opportunities that are now more available than they used to be. Things like the data, more about that, and biobanks, geospatial data, the tools like databases and training. So you will hear about that as well. And then team science, of course, is absolutely, it has to be team science or it's not going to work with all of these different kinds of exosomics factors. Okay, and to skip to a few examples, many of these diseases you think about in our NINDS mission space are being studied within the exposome. This is just showing an ADRD, that yes, it's genetics and yes, it's exposomics, but also time is very important. And the exposome really starts in prenatal life and follows through early adolescence and into adult life. And actually, most of the epigenetics happen early on. This is just showing with Parkinson's disease, an agenda that includes environmental factors, toxicants, microbiome, et cetera. And then also even things like spinal cord injury. In this case, it's a simple environmental enrichment cage in rats, increased regeneration of the dorsal root ganglion and neurite outgrowth after nerve transection. So you can think about what this could do in humans. In this review, they show many exposomic factors, and this is for cardiovascular disease like smoking, air pollution, bad water. And the important part is what kind of work like this can do for future health research as well as preventative medicine. And again, as I was saying before, this is growing really. It's remarkable how much of this you can see now in the literature. These are just some multifactorial analyses. For this one, there was 348 environmental variables from the ABC study, and it was reduced in an iterative process of exploratory factors and analyses. And then they ended up with 6 of these factors that seemed to make the most sense. This is just one on neuroimaging, exposomic neuroimaging, and basically brain imaging phenotypes were determined by genetic variations with environmental exposures, gene-gene interactions, environmental interactions, gene-environmental interactions, and basically the brain imaging phenotypes result in individual differences in cognitive abilities. So we actually did some work. I was mentioning before, we started some work, and the science with several funding opportunities, both on our own and some with other institutes and centers. And these so far include things like environmental toxicants, climate change, biopsychosocial factors, environmental stress, and how that influences health inequities, microbiome, some pain research, and so forth. And we have a couple more that'll be out soon. And with that, I think finally, to get started on our workshop on tools and resources, I wanted to just show a picture of the kinds of data and analysis tools, and in this case, described by a very strong group overseas. I'll say there's a lot of exposomic research going overseas. And for example, starting with the tools, then the data, and then finally the translational part for interventions, which is extremely important. And we're not doing this if we're not going to be looking for interventions. So I think that is it. Thanks to our office, we have 3 branches, and I've focused on the exposome branch. I have a fantastic small group, but you can imagine it's not surprising for the exposome that we work with all of the staff at NINDS and many other institutes and centers, as well as other agencies. So that's it, and I guess if we have time, take a question or two. Thank you.
All right, we have some questions coming in now. So I can read them, and then Dave, you can answer. So the first is, can you share of any specific activities that you know about that may be going on overseas? So I guess not in the United States.
There's a lot going on. They've got pretty large cohorts that they're working with, and just looking at a lot of the different kinds of exposomic kinds of studies. There's a couple, I think, that are working with here in the States, and maybe when we hear about NIEHS really started this, they may have some information on that, but I can say that it's definitely a lot going on there, because we've had some meetings with some of them. So yeah, I just don't know specific right now.
Okay, but certainly there are, international societies that can be focused on the exposome as well. So, okay. The next question is, are there studies that you know about that relate, I guess, that investigate the exposomic factors that underlie autism, or that may relate to autism?
No, I don't know a lot about autism. It's sort of not one of the things that especially in our mission space, that one of the things that we've done. But I'm sure that there's some studies going on. I would not imagine, there's a lot of studies going on, and I would think this would be one of them. So I don't know the answer to that. Sorry.
Okay. The next question is, if you could expand on the impact of the exposome on racial and ethnic disparities.
Right. Yeah. Well, I mean, you can think about some of these, areas where there's a lot of pollution, and that pollution probably increases the stress. And when you have that stress and you have the other kinds of factors, I think that's probably a major part of some of the things that you see in those areas.
Okay.
And we've got one that we've just finished on that stress part, that yeah, we've just finished on that, and there should be some studies coming out soon.
Great. How about I'll see. Okay. Well, the questions are now coming thick and fast. Excellent. So how is climate change affecting the exposome?
Oh, Well, it's when you think about heat and you think about people who work in heat, um, that's a major factor in health. So, there's lots of studies that's looking at that right now. So yeah, I mean, I right now we've maybe got one study going on, maybe another one coming soon, hopefully. But yeah, I mean, it's bottom line is it's that we know that heat is not healthy. So
Okay. The next question. I see that research was done using mice. Do you believe data will still be the same if you were to use Drosophila melanogaster, fruit flies? If not, what else could you test in the studies besides mice?
well, I mean, it depends on what you're looking at, and um, the specific kinds of things that you're looking at are going to probably be related to the kind of what you'd want to work with. So it just depends. At least that's what I would think.
Okay. Think I missed this question earlier, but uh, can people reach out directly to request advice on submissions?
Sure. Yes. We always, encourage people if they want to talk about specific areas of research, give us a call.
Okay. Um, that one's
Uh-oh, I've got to turn
yeah.
this off. 1 second. It might be good to move on because this person's going to 1 second.
Oh, Tuck, you have your hand raised? Oh, sorry. Hold on. We can't hear you. Tuck, can you unmute?
Okay.
Yes.
I wanted to add a very brief comment to David Jett's remark about unhealthy aspects of heat. Ozone also increases directly as a linear function of heat over the 10-degree projected range. So it's not just the thermal effects, it's that we have to contend with. It's increased levels of ozone on a global basis.
So how are we doing on time?
We're at time.
Yeah, and please, I'll try to answer questions in the chat when I can.
Okay. Thank you, Dr. Jett. So yeah, at this time, we'll move to the next presentation. And that will be a presentation on some of the resources at NINDS. So the next talk will be given by Dr. Rebecca Price and Dr. Chris Swanson-Fisher. Dr. Christine Swanson-Fisher is a program director in the Division of Neuroscience. Among her current duties at NINDS, she manages a portfolio of research grants and cooperative agreements and supports NINDS clinical data and biomarkers-focused programs in Parkinson's disease, including the NINDS PD Biomarkers Program and the Public-Private Partnership, Accelerating Medicines Partnership for PD. Dr. Rebecca Price is a program manager in the Division of Neuroscience. She oversees NINDS resources, including the NINDS Human Cell and Data Repository and the NINDS Biorepository, the Biospecimen Exchange for Neurological Disorders. Their talk is titled NINDS Biospecimen Cell Tissue and Data Resources. Rebecca.
Okay. Can you see the slides? Hear me okay?
Both.
Awesome. Well, thanks for inviting us to give this talk today. We're going to talk a bit about NINDS, biospecimen cell tissue and data resources, and we'll touch a little bit on the end on some other NIH resources we thought might be of interest to this group. So let's move on. Quick disclaimer and disclosure. And then we'll jump right in. So the first resource I wanted to talk about today is the NINDS Human Cell and Data Repository, or NHCDR. This is our biobank for storing patient-derived or healthy control-derived cell lines, fibroblasts, and induced pluripotent stem cells. So these are all lines from people that have genetic mutations related to those diseases, and some sporadic cases as well. And we have some edited lines. In this case, the edited lines that we have are taking out those mutations from the patient lines. You can get more information and details at the website, which is linked in this QR code. But our repository both banks and distributes cell lines. So if you are thinking about doing a study using human subjects, you want to collect PBMCs or skin biopsies and have those lines, have those cells turned into iPSCs, that's something that NHCDR can help. So you can reach out to your program directors or to me directly about how that happens. But the repository will generate those lines, and then all lines are available for broad distribution. So I have listed here the diseases that we currently have lines available for in the catalog. You can see a number of neurological disorders, neurodegenerative disorders, they're all neurological, and movement disorders, and we have control subjects. We also have some research grade iPSC lines from control subjects that are available. To access the lines, should be pretty straightforward. You go to our website, which is the catalog that houses all of the available cell lines, and it includes the available clinical and genetic information for each cell line. So you can browse through, see what we have, what mutations for those patient lines, information about age, gender, that type of information, and put them into your cart, and then you would set up a purchase order. So all of the lines do have a distribution cost, and that distribution cost we use to then put back into the repository to regrow these cell lines and keep the lines available for other researchers. We distribute globally to both academic, nonprofit, and industry investigators who are interested in using the lines for a variety of research questions. Okay. Next up is the Biospecimen Exchange for Neurological Disorders, or BioSend. This is our NINDS funded repository for biofluid samples. So I have listed here, again, like with NHCDR, we can both bank and we will distribute cell lines. So if you're running a clinical study, thinking about putting in an application for NINDS, banking with BioSend is an idea. And there's definitely a possibility and something to think about as you're submitting your application. So if you're interested, again, you can reach out to your program director who can help you get to the right people. You can reach out to me, or BioSend on their website has a form to request a quote. So you put in some information about your study, they'll send you a quote. We do charge for some kit and shipping fees at the front end, but then the storage and distribution of all of the cell lines are covered by the repository. So right now I have what our available sample types are, but we certainly collect things beyond what is listed here. So we have some ongoing studies. I don't have them there because they're not available for distribution right now, but we are working with collecting stool samples, and we have been in discussions about other types of tissues as well. So if there's something you're interested in, please reach out. We might be able to help accommodate that. What I have listed here for the disease areas are current disease areas that are available for distribution. But we have new disease areas going to be added to the collection soon, and then new cohorts within these disease collections, particularly within our traumatic brain injury collection. To get samples from BioSend, unlike NHCDR with the cell lines that we can regrow and maintain those lines, all of these, plasma CSF samples are a limited resource, so you do have to go through a review process. So we maintain several biospecimen resource access committees, or BRACs. Right now we have several BRACs that are centered around a disease focus. So what you would do is go into that disease area's catalog, determine the sample types that they have, the amount and availability for those diseases, and then you would submit an application via an online web form. It's a short 4-page application, including biosketches and information about what samples you want. Then we have 6 review meetings per year per BRAC. So we accept applications on a rolling basis. The BRAC reviews your application and makes a determination. And then we have sort of a post-BRAC process to help onboard you, help you select your samples, work with the repository to get everything shipped to you. Everything is shipped blinded, so adds a little extra layer of rigor there. And then once you've done your analysis, that data is sent back to us and we unblind everybody. So the data, all of the data from these samples gets shared back to the repository where the clinical data is, so that the data is then available for people for future analysis as well. If you're interested in submitting an application for a biosample request, you can email us at this link here, or email here. I'm also going to talk about a couple of resources. So I manage BioSend and NHCDR, but we have a few other resources that we offer as well. So one is the NINDS Human Genetics Resource Center. This repository houses DNA and lymphoblastoid cell lines. They have genomic data in dbGaP. They have things collected using common data elements, which you'll learn more about later in the webinar. And this is, again, another central repository for genetic materials. Right now, this is what is available in the catalog. You'll see there's a little bit of overlap with some Parkinsonisms and some motor neuron disease, but this collection also has some other diseases that we don't have in our other cohorts, like cerebrovascular dystonia, epilepsy, and Tourette syndrome. And information about the catalog and how to access these available resources you can find at this link here in that nice red colored highlighted. NINDS is also part of the NIH Neurobiobank. The Neurobiobank aims to facilitate science through kind of democratizing and modernizing how brain tissue is banked and accessed, including more standardization in the tissue processing and characterization. And they also work to increase brain awareness among diverse populations, so trying to get more people from more diverse backgrounds to donate their brains for research. It's funded by multiple ICs across the NIH, including NINDS, and involves subcontracts to 6 brain banks from across the US, at Harvard, Mount Sinai, UCLA, University of Maryland, University of Miami, and Pittsburgh. For more information about the Neurobiobank, they also have a website. So I'm showing here their page about obtaining tissue. Similar to BioSend, applications are required to get access to the tissue types, but you can go to their website, get information about what types of tissues from what types of donors they have. And there's lots of other information about how to access what types of tissues, requests, and publications there. So link there and also in the QR code.
I'll just note, Rebecca, really quickly. So these are disorders supported for the NIH Neurobiobank, not just with the NINDS mission. As Rebecca noted, it's a collaboration. So there's also disorders within the NIMH mission as well. And we also work with the NIA. And so it's not just disorders that are represented within those institutes, but it's also control tissue that's available from postmortem brain as well as spinal cord tissue.
Thanks, Chris. Okay, this slide has a lot of information, but we had a limited amount of time to talk about it and didn't have time to talk about all of the NINDS supported data resources. So we're moving away from those actual cell tissue biofluids into the data resources. So we have a number of data resources that are disease area focused, such as FitBur for TBI data, MAP MECSF, which houses data for myalgic encephalomyelitis chronic fatigue syndrome. The Parkinson's disease biomarker program houses all the clinical data associated with our PD samples. And AMP PD also has a knowledge portal with, Chris can give you lots of information about what's housed there, but it's a lot of the genomics analysis being done through AMP PD, which through this public-private partnership. So you'll have some PD samples, but also lots of other partners involved in contributing data to AMP PD. HEAL, or the Helping to End Addiction Long-Term, also has their own database. HEAL is really focused on opioid substance use and pain, and trying to develop non-addictive pain therapies. MARC VCID is a clinical consortium for vascular contributions to cognitive impairment, and they also have their own website and biospecimen collections. All FTD is jointly funded by NIA and NINDS and houses data and samples from frontal temporal dementia. Then we have a number of other collaborative sort of resources. So the Enigma Consortium brings together researchers in imaging genetics to better understand brain structure, function, and disease. So this is an opportunity to get involved and collaborate in sort of a web-based space. Data from NINDS supported clinical trials is also available. You'll see that at this link over here under NINDS for information about how to get access to those clinical trial data. The RDCRN is the Rare Disease Clinical Research Network, has a data management coordinating center. RDCRN has 20 different networks and consortia focused on different rare diseases, including but not limited to neurological disorders. For example, one is the CREATE Consortium, which is an ALS natural history study, but you'll find a variety of other data available there. The Precision Medicine platform is a cloud-based system that allows researchers to collaborate and analyze large data sets in a cloud-based environment that's secure and using a lot of machine learning power there.
Yeah, we have a number of our stroke clinical studies that are leveraging the Precision Medicine platform as we've been working together to do curation and harmonization of a number of our NINDS and non-NINDS supported studies in stroke. So that's just one example there.
Thanks. And then we wanted to highlight a number of different resources available through the BRAIN Initiative. So BRAIN Initiative celebrating their 10 years. So this is a trans NIH initiative to understand the brain and map atlases. So highlighting a few resources here. The Cell Census Network, BICCN, is a comprehensive reference of diverse cell types in human, mouse, and non-human primate brains. The Brain Initiative Cell Atlas Network, BICAN, is a collaborative effort to create a comprehensive atlas of the human brain. And the Neuroscience Multi-Omic Archive, NEMO, is a data repository specifically focused on the storage and dissemination of omic data generated from Brain Initiative projects. The BRAIN Initiative also has several dissemination cooperative agreements in this U24 program. So there's more information there of things to get involved in. And then finally, I wanted to highlight some other NIH resources that are available that are not funded by NINDS but might be of interest. So certainly not a comprehensive list of what's available at NIH, but some things that we thought might be of interest. So National Institute of Aging has several resources. NICRAD, our sister biorepository, houses biofluid samples from Alzheimer's disease and related dementias. And the National Institute of Aging Genetics of Alzheimer's Disease Data Storage, NIAGADS, also has genetic information from Alzheimer's disease studies. There's also the Excel AD, exposomics for characterization of environmental drivers of AD community dashboard, and the AD knowledge portal. The NIH Common Fund, which is helps support things across all of NIH. So again, very diverse types of data that you'll find in the metabolomics workbench and the Common Fund data ecosystem. NIDDK, the National Institute of Diabetes and Digestive and Kidney Diseases, also has their own central repository for data and samples. And there you can probably find a number of things that might be of interest if you're looking at metabolomics and gut microbiome studies. And then finally, we wanted to highlight the iPSC Neurodegenerative Disease Initiative, or INDI, which is an intramural program to develop iPSC lines in neurodegenerative diseases. So it's sort of a complementary effort to our patient-derived cell line repository. INDI takes all isogenic lines and is using CRISPR to edit in mutations of interest for neurodegenerative diseases. So I know that's a lot to digest, but I know the presentation will be saved. So hopefully you can come back and grab anything that you are interested in.
And we'll also add these links in the chat as well today through the presentation. Thank you.
Yes, thanks. And we hopefully have a little bit of time for questions.
Okay. Okay, thanks, Rebecca and Chris. Thorough. I have a question while people are kind of typing away into the chat, so maybe this will get us started. So, we saw across all of your slides a lot of I mean, there are a lot of resources, and they're spread, around NINDS and around NIH. So is there a single place where people can find kind of like the full list? And I guess maybe the corollary is, how does one know which is the right resources? I mean, it's almost exhausting to go through the entire list. And this is, I think, just at NIH, I should say. And of course, there are, data resources and sharing and banks that are not maintained by NIH, but by, nonprofit partners and what. So what would you recommend?
Thanks, Neil. This is a big challenge, and something I know we've talked with a number of people, around NIH and outside of NIH. So we try our best to make things available. Some of the information about our repositories are available on our website. Not always the easiest place to find, but we can certainly direct people that way. But it's something we've been talking about and trying to figure out. These types of talks, I think, are a great way to kind of bring stuff together. But if you're not attending one of these webinars, if you don't know that it's happening, it can be daunting to find the different resources. I guess
I always say, reach out to your program officer if you haven't. If you are interested in submitting an NIH grant, specifically within the NINDS mission, reach out to us. Many times we're talking to each other. we're also within NINDS. We have a data science a working group as we're working together to implement the new data management and sharing plan. We've set up a lot of materials about where is best repository to deposit your data, or repositories potentially, and then are there privacy linkage, such as global unique identifiers to be able to link across. So we have a lot of those resources. As far as sharing across non-NINDS funded resources, I work really closely with my colleague Kemeny Riley here at ASAP, also our colleagues at the Michael J. Fox Foundation. So for instance, the PD BRAC that Rebecca mentioned, we share that with the Michael J. Fox Foundation for some of those cohorts. And Michael J. Fox Foundation and NINDS and ASAP are all part of the Accelerating Medicines Partnership in Parkinson's disease as a way to democratize the data across all of these research areas, to enable broader access to the wider research community and the entire community as well. So
yes,
a number of different strategies we have.
Yeah, and we're starting to do similar things across the other biospecimen resource access committees. Like we'll be trying to federate access to data sets and biosamples that are available at other federal agencies, in working together to kind of harmonize efforts that we're doing in new initiatives, to make sure that we're not collecting the same information across studies. So there are efforts happening, and we try our best to kind of coordinate all of those pieces together.
Okay. Thank you. There's also a comment in the chat. In some other areas, I guess people put together metadata directories to help.
That's excellent.
Okay, so here's one question. Which repository has blood or cerebrospinal fluid from sports players, military personnel, or people that are exposed to pesticides, like farmers? Not necessarily people with a particular disease, I guess, right? So these are like repository related to the exposure. The assumption is that people are exposed for years or decades before any disease process starts.
So FitBur would be a good place to start for military personnel. It is very focused on traumatic brain injury, but FitBur is a collaboration between NIH and the Department of Defense, the research branch. In terms of sports players and blood and CSF, we don't have that now, but there are some ongoing studies looking at TBI and CTE in people who play competitive sports. So we will have some of those samples soon. That data will, again, all be in FitBur as that sort of TBI, CTE repository. In terms of
There
I
are
think-
also some
Mhm.
NINDS repositories. So obviously, Boston University has some of this data, both ante-mortem and post-mortem, as well as a Million Veterans program as well has this data. Although as it's VA, there are stringent capabilities for adjudication of access to those biosamples as well. But great question, Colette.
Okay,
In terms of
thanks.
people just exposed to pesticides, I don't know of anything that is that broad, but I think some of these, projects, and maybe we'll hear more from our colleagues at NIEHS in a talk later today about where those types of resources might be available.
Yeah, but maybe I can follow up on that. So on, like, for example, the existing Parkinson's related biosamples that we have at NINDS, do those track things like pesticide exposure?
Yes, so we do have some common data elements that are tracking as far as lifestyle exposure, and that's something as well that we'll hear a little bit more about exposomes for CDEs, and it's a growing area of interest, not just within Parkinson's, but across the NINDS mission. So that's a great question, Neil. I also saw somebody ask a question about sample analysis services. So there is a possibility for that. And I think Rebecca, if you want to put your email in the chat, then I think you could probably follow up offline with this user about the additional details there. Another great question.
Yes, will do.
I see
Okay.
Kulip has his hand raised. I don't know if
Oh.
Kulip, did you want to say something?
Attendees are unable to speak for this meeting. We will need them to put their question in the Q&A.
All right,
Okay. And Kulip, we're happy to also connect offline too. Thanks.
Our next speaker is Dr. Yusha Cui. She is a health scientist administrator at the National Institute of Environmental Health Sciences. Dr. Cui oversees the exposure science and the exposome grant portfolio that is focused on emerging technologies towards improved exposure and health impacts assessment in environmental health research. Dr. Cui currently serves as the co-director of the Human Health Exposure Analysis Resource, a trans NIH program that supports a centralized network of exposure analysis services and expertise to enable comprehensive environmental exposure assessment in population health research. Her talk today is titled the NIEHS Exposure Science and the Exposome Program, Creating Tools and Resources for Exposomics.
Thank you, Neil, for the introduction. Okay, as Neil mentioned, I'm a program officer at the National Institute of Environmental Health Sciences. And the title of my talk is the NIEHS Exposure Science and Exposome Program, Creating Tools and Resources for Exposomics. Just a disclaimer saying this presentation does not reflect official policy of NIEHS and NIH, and I have no financial interest to disclose. So most of my talk today will center around the research activities at NIEHS to support exposome research. This includes the Human Health Exposure Analysis Resource, the Comparative Toxicogenomics Database, and also a new trans-NIH initiative for exposome research coordination. But before talking to these specific resources, I want to take a step back with you to think about what tools and resources are needed for exposome research. So the exposome, as mentioned by Dave, it's a totality of exposures that an individual is subjected to from conception to death. And the study of the exposome called exposomics. In last year, December, a group of experts around the world got together to try to develop narratives around what is exposome research, or what is exposomics, or you can see that's the operational definition. The group came up with a definition says exposomics is a field that studies the comprehensive and cumulative effect of physical, chemical, biological, and psychosocial mediators that impact biological systems. By integrating data from a variety of interdisciplinary methodologies and streams to enable discovery-based analysis of environmental influences on health. They specifically pointed out that exposomics is not a single implementation, and it can be adapted to the specific system and the questions being asked. For instance, when we talk about neural exposome, in my mind, that's implementation of exposomics in nervous system health. And we are asking the question, how does exposome impact nervous system health? And it can be other systems in other disease areas. And that's kind of my understanding of exposomics and how it's related to the specific missions of different ICs and disease interest. So that we talked about the kind of operational definition, but how to operationalize exposomics? How do you conduct an exposome study? In 2022, the NIHS organized a series of virtual workshops during the summer. It's called Accelerating Precision Environmental Health, Demonstrating the Value of Exposome. So the workshop series attracted a large number of participants, and with many hours of Zoom meeting discussions. And there were a total of 64 breakout sessions discussing topics that the participants think that's important for their research. And we ended up with more than 200 pages of meeting report to report out the discussions of these 64 breakout sessions. And of course, all these issues discussed kind of interrelated, and some kind of repeat of each other, or kind of from looking at one question from different angles. This diagram essentially summarizes all the discussions. It was summarized as 10 priority topics in 5 areas. The 5 areas can essentially be seen as kind of representing the kind of the key steps of exposome research, from left to right, thinking about what to measure and how to measure, and what's the method available to get the information that we want. And there's also discussions around how to share and harmonize data. And of course, downstream, how do you integrate the data and analyze, interpret results, and translate the data into public health intervention and more. So I hope, for the rest of my talk, when I talk about the resources, you can somehow keep this diagram in your mind, thinking about how resources help address some of the steps. And I would like to see the resources I'm talking today is mainly focusing on the kind of the steps on the left, more on the what to measure, how to measure, and how to share and harmonize. And we can discuss more. So the first resource I want to talk about is the Human Health Exposure Analysis resource, or called HERE. So this is a program under the bigger NIH Exposure Science and Exposome program, which focus on developing tools and technologies for measuring the exposome from external or environmental concentrations of exposure to internal dose to associated biological response. The technologies we supported including geospatial technologies, remote sensing technologies, or wearables to get personal exposure, or biomarkers of disease and exposure, or omics technologies. So the goal is to put all the tools together in an integrated way to apply to a study to enable the comprehensive and systemic exposure assessment. That way, we can get a more holistic view of what people are exposed to, and to get more accurate evaluation of exposure disease association, and help us to understand the gene environmental interactions and how that contributed to our disease etiology and progression and treatment. And here is one of those kind of infrastructure programs to enable the kind of the comprehensive application of different technologies. Here is was funded in 2019, and it was a continuation of the Children's Health Exposure Analysis resource. The goal of the HERE program is to provide an infrastructure for adding or expanding exposure analysis, kind of as a resource, to the community, to the NIH supported research, health research community. HERE consists of 3 components: a coordinating center to coordinate data activities within the consortium, and also it's out-facing of the program. It also consists of an exposure assessment lab network, and a data center, which not only stores the data generated by the laboratory network, but also the health outcome data associated with exposure analysis or the study we support. It's a program supported by multiple NIH ICs and offices. This is a kind of more detailed, a little bit more detailed description of what HERE offers. The program offers exposure analysis of both targeted, thinking about the CDC type of targeted by monitoring of a particular chemical or a group of chemicals, and untargeted, think about untargeted metabolomics that can measure both environmental chemicals and endogenous metabolites. And these methods can be applied both in biospecimens or environmental samples. And HERE also provides laboratory methods consultation, how to select the right method, and also sample collection, storage, and integrity, as well as epi study design. The data center serves as a data repository and also provide data science tools and statistical analysis. This kind of more detailed listing of the target analysis. If you want to measure, say, brominated flame retardants or DNA damaging agents or environmental phenols or nutrients, so there are specific methods associated with those kind of targets you want to measure. And there's also untargeted analysis of both environmental samples and biospecimens. And these methods are available on the HERE website as listed on the top. On the next slide, I know you can't read it, but so this is a HERE sample metrics table, which I think is really useful. On the top, it gives you, I'll make it larger so you can see it. On the top, you can see the different types of biospecimens, say urine or plasma serum or saliva, or environmental samples. And in this different type of matrices, think about what kind of exposure information I can get from this. For example, if you have, say, whole blood, you can get measurement of brominated flame retardants. You can also get DNA adducts measurements, and you can get a trace element measurements, which it's being cut off. So you can use that way. You can also think about, if you're doing a research, thinking about, okay, I'm interested in this type of exposure. How can I get the information? And you think about what kind of samples I need to collect. So it can work both ways. And using this method HERE has supported 53 studies proposed by individual researchers. This includes almost 90,000 specimens and almost 50,000 study subjects and many chemicals. The studies is listed on the right across a wide range of health outcomes. So this provides the opportunity to kind of compare study. If you, for example, if you're interested in asthma, there are 16 studies measuring different things, and put together, you get a bigger picture of, okay, if I'm interested in studying asthma, what kind of environmental exposures I should consider, and based on what others have found, that give you some hypothesis generation opportunity. And I want also to add that those are just studies proposed by individual researchers, but the HERE program also supported some other NIH programs such as ECHO, CARE, All of Us project. The data is not included here, but it will be shared through the program portals or repositories. The next, I'm going to briefly introduce the comparative toxicogenomics database. So this is a project has long support from NIEHS, and now it's also supported by NINDS. The PI is Carolyn Madeline in North Carolina State University. The goal is to understand how exposure to environmental chemicals affect human health. It was first launched in 2024, so 20 years of data creation. It's user-friendly, no login passwords or accounts needed, and manually created data from the literature. And it's a CTD provides or create a relational database that links 9 data types that include organisms, genes, GO phenotypes, exposures, disease, pathways, chemicals, and it enables computational predictive network modeling and also empowers users to generate testable hypotheses. For example, I think at the beginning when the audience asked, what kind of studies have been done for autism. For this database, you can put in autism as a disease or phenotype, and then you pull out all the relevant genes, chemicals, pathways, and also a variety of organisms, model organisms has been used to study this. So it's a great resource for hypothesis generating. And the CTD has a large database. There's more than 2 million chemical gene interactions created. There's almost 400,000 phenotype-based interactions, and there are lots of gene disease associations or chemical disease associations. It's a great resource. So last, I'm going to talk about the Trans-NIH Initiative for Exposome Research Coordination. I want to go back to the diagram we initially talked about. We talk about how CTDs can give you some ideas about if you're interested in a particular disease area, what to measure. And the HERE resource is a place maybe you can find, well, if I'm interested in some of the chemical exposures, what are the methods available and what kind of biological or environmental samples I can collect to get that kind of exposure data. I did not talk about, but the HERE actually the HERE program also we have data science tools, ontologies to help with data harmonization and data sharing. If you remember what Rebecca and Chris presented as some of the data sources, the database is already available there. So there's a question about how to merge the data from different data streams, including some existing databases and towards the question you ask. But the available resources obviously first is not meeting all the needs. Second, as the audience already asked, it's all over the place. how can I get all the resources together to serve my purpose? So as a community, we really need that kind of work. So that's the reason we are going to establish a Center for Exposome Research Coordination. This is going to be a program to be funded in the summer soon, in a few months. It is supported by NIHS, NINDS, NINDS, NCI, and Office of Research on Women's Health. The center will also work in concert with some of the EU efforts, the International Human Exposome Network. Some audience asking, what's the efforts happening in Europe or other countries. So we hope the program will work with the major efforts in the different places to coordinate the resource and tools. And that's kind of a picture in our mind. We hope the US program we're going to fund, we're going to work with the European programs along with other efforts around the world for coordination of resources and tools. And that's all I have.
All right, thank you very much, Yusha. We have time for a quick question, maybe two, if they're very quick. Okay, well, if you do have questions, you can ask them in the Q&A box, and we can answer them asynchronously or bring them back up in the general discussion at the end of the webinar. Now we'll move on to the next talk. Next up is Dr. Timothy J. Buckley, the Senior Science Advisor for Exposure Characterization and Modeling in ORD's Center for Computational Toxicology and Exposure. He previously served as Acting Associate Director of Health and Ecology within EPA's Center for Public Health and Environmental Assessment, and a Division Director within the National Exposure Research Lab. His work is broad-ranging and includes the development and application of exposure methods, measurements, and models to chemical, physical, and biological stressors within community and occupational settings. Dr. Buckley's talk is titled EPA's Computational Exposure Science Tools and Resources on a Path to the Neural Exposome. Dr. Buckley.
Yeah, thank you very much for that introduction. And thank you for the invitation to be a part of this workshop. There's some work within the agency that I think is very much relevant, so I'll move right into that. A couple of caveats. Number one, these are my views, not those of the agency. And secondly, I don't have any financial conflicts of interest. And just by way of context, you know, the research and development that occurs within the agency is oriented toward in support of the program office as a regulatory agency. So with risk assessment being the scientific foundation for supporting policy, much of our research is risk assessment oriented. So exposure science is very prominent within the agency. I would say not so much the exposome. But as I depicted in the Venn diagram, I think there's a lot of overlap between, you know, where it is that we're headed with the exposure science and what is of interest to the exposome. And I'm going to touch on just two examples of those research activities. The first has to do with cumulative exposure, and the second has to do with comprehensive consideration of chemical exposures. So first of all, with respect to cumulative exposure, there is great interest within the agency in this area. You know, I think I really like the way that Vermeulen in his Science article in 2020 kind of characterized the exposome and its comprehensiveness in considering, ecosystem, social, physical, chemical, and lifestyle, very consistent with, I think, Dave Jett's depiction also of what needs to be considered in the exposome. Nicole Tolve recently published an article in Jesse that kind of provided that same kind of characterization, depicted slightly differently. But there's very much awareness within the agency of the importance of the comprehensiveness with which we need to consider stressors in the environment, both stressors that are harmful, but there are also salutogenic stressors in the environment that need to be taken into account as well. All right, so the two examples are taken from the published literature. The first one has to do with green space, environmental justice, and depressive symptoms. Obviously an important outcome in the context of this workshop, but also importance because of the disease burden with something on the order of 18% of US adults reporting having depressive symptoms. So and then the second example has to do with the computational chemical exposure research activities within my center, which I think also are relevant to the chemical exposome as might be considered in this workshop. So just in terms of this first research activity, again, I think the relevance here is because of kind of the comprehensiveness of consideration given to lifestyle, ecosystem, social and physical and chemical. So all of those aspects were kind of taken into account in the context of this study, which was a collaboration with NIEHS and their sister study. So we accessed a subsample from this larger cohort of 50,000. There was some 3,555 individual women across the United States that were in cities that had previously been historically classified by HOLUC or redlined districts within those cities. So the home ownership loan corporation classified different neighborhoods as A, B, C, or D, and so that was taken into account into this analysis, along with the depressive symptoms that were reported from the sister study using this CESD 10 scale. Let's see, which is shown here. And also taking into account the tree canopy within 500 or 2,000 meters. We considered both of those buffer zones, as well as climate zone, and as I mentioned, the redlining within those cities. Because we had individual level data, we also took into account individual level demographics and things like medication use for depression. And all of this data was analyzed using a mixed model approach. And I guess maybe not too surprisingly, we observed the increased odds of depressive symptoms in lower HOLUC grades. So kind of in this stair-step fashion with the redlined or the grade D neighborhoods, we observed the higher rates of depression. And similarly, we saw kind of this stair-step decrease in tree canopy within each of those successively graded neighborhoods, with the grade A being the highest income, and grade D, the lowest or the redlined neighborhood. With as much, on average, 20% more tree canopy within the grades A, B, and C relative to D. So then, looking at depressive symptoms as an outcome within the full model, we saw a significant effect associated with tree canopy and the redlining, and so the whole the grade had an effect along with the tree canopy. We were able to tease out those effects within the mixed model, and we saw that the redlining had the effect was not linear, with a stronger effect within the grade D or the red line neighborhoods. So, so tree canopy was, less within those neighborhoods, and it had a stronger influence on depression within those neighborhoods. So that's kind of the first example, kind of that I think is important in terms of the comprehensiveness with which exposure was considered. And the other thing I like about this is that it's not only looking at the adverse effects of the environment, but in this case, it's looking at how it is that human health and an important, neurological outcome of depression is influenced by, in a positive way, by something, associated with nature of tree canopy. And this was published in Environmental Health Perspectives a couple of years ago with the lead author of Wei Lun Tsai. The second example I'd like to talk about has to do with chemical exposure, or the chemical exposome, and its importance to public health. And I think this article from Landrigan, although it's a little bit dated now, really highlights how significant chemical pollution is to public health, with some 9 million premature deaths estimated in 2015, and he also talks about the importance from an environmental justice standpoint. Those that are least able to withstand the health impacts are the ones that are receiving the largest burden of pollution. And of course, the linkage to climate change. So pollution, it's not just pollution, but it's also feeding into the issue of climate change. And we know that chemical pollution has this large impact on public health, but we, for the most part, don't know how that works, what it is about the exposome, the chemical exposome, that is resulting in that morbidity. So I think very consistent with David Jett's article in Neuron, where he kind of talks about the neural exposome. There's just a lot that's not known about what is contributing to these neurological diseases. And so Rappaport in 2016, I think identified the importance of non-genetic factors for a variety of chronic diseases, but among those were very important neurological diseases, some of which others have talked about already, like Parkinson's disease. And so I think this is important because it's not at all about the genetics. We're not going to be able to solve these diseases with a focus solely on the genetics. We really do need to take into account the exposome and the chemical exposome, I think, is particularly important. And so you can see a number of important neurological diseases here that are represented by the genetic population attributable factor or fraction. And so with the genetic fraction identified here, the balance of that is the environmental. So the bulk of the influence in these chronic diseases is associated with the environment. So with that kind of background and rationale, within the agency, we have a very expansive research program in trying to assess the exposome, and within my center, this is characterized as a computational exposure. And again, I think it starts to get us moving in the direction of the exposome. And so there's just a couple of things that I think are important as we go down this path. You know, one is the incredible complexity associated with the exposure or exposome, not only from a chemical standpoint, but you take into account all of these other social built factors that come into play, and it becomes very complicated very quickly. And just the number of chemicals that are out there in a modern society make this an incredibly challenging endeavor. So we need lots of data, and we need models. This is not it's not an effort that we're going to measure our way out of. We need a combination of the data and the models in order to kind of provide estimates for this exposome. The modeling takes on a really important role because the number of chemicals are so vast that we just cannot measure our way out of this. And we're looking to be predictive, rapid, high throughput. Yeah, this information needs to be provided in a timely fashion. So it's this computational exposure effort, which was described by Peter Agegi in 2016 as the integration of advances in chemistry, computer science, mathematics, statistics, and social and behavioral sciences to create these data and models. And that's really what I'm going to talk about, is kind of an update as to where we are in these research activities. And so the way that the research is kind of organized across chemical curation, data development, modeling, and then these cross-cutting considerations. And the way that I view this is that the chemical curation is absolutely foundational to the subsequent efforts, the data development and the modeling. I view the chemical curation as kind of like the lifeblood of the computational exposure efforts. It flows into everything else. The data development is like the appendages. So that's gathering available information that's relevant to chemical exposures, whereas the modeling is the brains. That's really kind of tying everything together and making interpretations about the exposome. And then the cross-cutting, that's like the personality associated with all of these efforts that allow the information, the estimates, to be delivered in a way that is friendly and trustworthy and inspires confidence among decision makers. So I'm going to take each one of these activities kind of in turn and just provide a very quick overview of them. The first one is the chemical curation. You probably all are familiar with this, the COMTOX dashboard. So in my mind, this is the most comprehensive accounting of the anthropogenic chemical landscape. So the agency has curated some 1.2 million chemicals now within this database. It's highly quality assured, includes lots of metadata, including the chemical identification, the structure, and all of this data is made publicly accessible and accessible in a way that is suitable for computational purposes. So it's absolutely foundational to the data acquisition efforts as well as to the modeling efforts, in a very critical piece of the puzzle for our chemical exposome work. All right, the next major kind of effort has to do with the data development, that is acquiring the data that's relevant to informing the exposome. And you probably all are familiar with the source to continuum framework. And just to point out that this is the framework by which we are populating data to inform the exposome. So it's information that's related to, on the one hand, chemicals that are in various sources or released from various sources. And on the other hand, the receptor, the human and the behavioral, physiological factors associated with the receptor that bring humans into contact with chemicals in the environment. Excuse me. So that continuum provides the framework. The way that data has been accumulated is very much consistent with the guidance that the National Academy of Science has given us in terms of its organization and usability and access. We're using very much advanced methods of informatics, data mining, and machine learning in terms of acquiring and organizing that data, and again providing public access. It's very much receptor oriented. We rely heavily on NHANES data to inform the source to receptor model or modeling. And so a couple of examples of the kinds of databases that are available as a part of the data development effort include ChemExpo, CPDAT, which is this data resource that pretty much defines the chemicals that are found in consumer products, some 10,000 chemicals. MMDB, a very comprehensive database of existing measurements across a wide range of environmental media. And we're moving in the direction of occupational exposures. And a part of I'm going to just highlight. Yeah,
Dr. Buckley?
Yes.
I'm sorry to cut you off. I think we're at time. Do you want to maybe jump to a conclusion?
Yes, thank you. I didn't see you pop in, so let me just jump to the summary slide. So in summary, the exposure science is grounded in the agency's risk assessment needs. I provided a couple of examples of how it is that we're kind of moving in the direction of the exposome, cumulative exposure, tree canopy, environmental justice and depression symptoms, and this computational exposure effort. So a number of other people were involved in this research. And my contact information and references. Thank you very much.
All right. Thank you very much, Dr. Buckley. We don't have time for live questions right now, but we can revisit them in the general discussion, or they may come in through the Q&A box. Sorry to have cut you off at the end there.
No problem.
Okay. So moving along, our next speaker is Dr. Valerie Adams, a senior biologist with the Defense Health Agency, Defense Centers for Public Health, Aberdeen, Toxicology Directorate, formerly Army Public Health Center. Currently, she conducts in vitro testing to support warfighter and civilian occupational health and environmental safety, and manages the Health Effects Division Toxicity Assessment Program. Dr. Adams is the president of the Defense Technical Information Center, End Users Council, and the chairwoman of the DoD Tri-Services Toxicology Consortium. Dr. Adams will give a talk titled 80 Pounds of Light Stuff, Building a Rational Exposure Assessment Toolkit.
Okay, thank you very much for the opportunity to present information on some of the biomedical research and evaluation efforts that are underway at the Defense Health Agency. Next slide, please. So I have a list of disclaimers to include that the views expressed in this presentation are my own and do not necessarily reflect the official policy of the Department of Defense, the Defense Health Agency, or the US government. That the mention of any non-federal entity or its products is for informational purposes only and is not to be construed or interpreted in any manner as federal endorsement of that non-federal entity or its products. My review and summary of the congressionally directed medical research program and the toxic exposures research program, CDMRP and TERP, respectively, do not necessarily reflect the opinions and views of those groups. And all the material that I'm presenting today is publicly available and was sourced from the internet. Next slide. So my goal for this presentation is to provide some background on the DoD biomedical research and evaluation components, and to give some context for the research, testing, development, and evaluation, our TD&E, enterprise, and the associated environmental health and occupational safety programs, and the current needs-based funding mechanisms that are available, the research programs that are currently underway, and some of the challenges for exposome research. Next slide. So I'm showing a very simplified version of an organizational flowchart here. The Defense Health Agency was established in October of 2013. So we're new by the DoD to support the uniform branches of the DoD and to direct the execution of joint shared services, such as the military treatment facilities, also called MTFs, research and development, and public health. Additional reorganization and redefining of roles came in the 2017 National Defense Authorization Act and subsequent congressional guidance. Since then, the DHA has been consolidating and reorganizing the various assets. In 2022, my organization, formerly the Army Public Health Center, became the Defense Centers for Public Health Aberdeen. Likewise, the Navy and Air Force components each became DCPH Portsmouth for the Navy and Marine Corps public health components, and DCPH Dayton that represents the Air Force public health components. The medical research development components, which when you look at this slide, it's sort of center at the bottom towards the right. That group and those components are currently in the process of rolling into the DHA enterprise. And so one of the reasons this slide is kind of fuzzy on the details is because the names and the way that those organizations and individual components are interacting is currently under development. I would also like to point out on the left-hand side of this slide, under the acquisition and sustainment block, that there are additional programs referred to as CERDEP and ESTCP. And in tiny writing, it says Strategic Environmental Research and Development Program for CERDEP and the Environmental Security Technology Certification Program for ESTCP. Those programs are responsible and support RTD&E for sustainable alternatives, conservation and mitigation and cleanup. At the Defense Center's Public Health Aberdeen Toxicology Directorate, We support both CERDIP and ESTCP, as well as the military research program efforts through consultation, toxicity assessments, and testing. Next slide. So I'm going to first focus on the occupational evaluation and research, and this is primarily what my organization works on. Similar, the RTD&E phases occur early in the acquisition process. So on the left-hand side of this slide, what you're seeing is sort of the acquisition life cycle from the original concept all the way out to sustainment and potentially disposal. And most of our work is where that arrow is pointing, early in development. Systems that are in that development pipeline undergo a rigorous environmental health and occupational safety evaluation. The Toxicology Directorate supports that activity. We use a phased approach to evaluate toxicity with the level of effort scaled to the budget activity. That's the BA. So on the portion on the right-hand side, you see BA1, BA2, BA3. That is a system or product that is working its way through those early acquisition stages. We support the toxicity testing effort at the sort of appropriate funding level and technology readiness level. Ultimately, these data that we generate are used to meet various acquisition and regulatory requirements. So what you see on the far right-hand side of that is the acquisition section. So we contribute if there are any National Environmental Protection Act criteria or other requirements through acquisition. Our work supports that effort. And what you see down towards the bottom of that between those two maroon triangles, milestone A and milestone B, again, that points you back to the figure that's on the left-hand side to see where we are in that early phase of development. Next slide. So on the operational evaluation and research slide, similar to the previous slide, the biomedical research efforts have technology readiness levels, or TRLs, with early novel concepts and emerging technologies in TRLs 1 through 3, applied research, translational research, and solutions for military problems in TRLs 4 through 6, prototype maturation and demonstration in TRLs 5 through 8, with production and distribution in 9. I'm emphasizing the structure of these TRLs because the funding mechanisms are targeted to specific levels. And it's important for people who may be listening that are interested in submitting funding to these lines of effort that they understand where the research is going to fit into this scheme. Next slide. So the congressionally directed medical research programs, CDMRP, invests in groundbreaking research across the full spectrum of research and development, including basic, translational, and clinical research. By strategically funding high-risk, high-reward research, projects that other agencies may not be willing to fund, the CDMRP addresses critical research gaps, and their funded research benefits not only military members, military retirees, and their family members, but the civilian population as well. Shown on the left-hand side there is just sort of an idea of how toxicology, exposure science, forest health protection, and risk assessment all kind of come together. There are 36 programs that are in the CDMRP, one of which is the Toxic Exposures Research Program, or TERP. Next slide, please. For TERP, some key points here. $30 million have been appropriated for this program. Their support is towards Gulf War illness, burn pits, and other airborne hazards, neurotoxin exposures, And then other toxic exposures in general, to include prophylactic medications, pesticides, organophosphates, toxic industrial chemicals and materials, metals, and potentially minerals as well. And the focus is on speeding development for treatments, cures, and preventions that are going to maximize the benefit to service members, veterans, and the American public. Next slide, please. So I took this slide from the 2022 stakeholders report from the CDMRP, and which is very thorough. There is a link at the end of my presentation for people that are interested in seeing the full report. This word cloud is a result of a request for information regarding critical issues, underfunded areas, and coordination and collaboration across other federal agencies. And as you can see, the prominent themes are poorly defined exposures, problems with monitoring exposures, access to samples and specimens, and a general lack of data, along with some of the other aspects there. Next slide. And then on the flip side, the recommended solutions for those gaps and priorities are shown in this word cloud. The respondents emphasized collaborative research and funding for foundational research. And at the bottom there, it says fund a diverse array of exposure. I feel like they probably left something off of that particular option. Exposure research, I guess. Next slide. So so that was for the TERP. So the Military Operational Medicine Research Program is also part of the CDMRP. And they have an environmental toxic exposure program, and their research objectives are shown on this slide. And the efforts include identifying and developing biomarker assays, improving and demonstrating technology for rapid screening of toxic chemicals and materials, and to provide health or toxicological data to fill data gaps for military relevant chemicals, materials, and environmental hazards. And note, you can see how that last one links us back to DCPH. And our environmental safety and occupational health activities, as well as how it might collaborate and cross-pollinate with the CERTUP and ESTCP activities and objectives as well. Next slide. And on this side, we just see how those objectives and specific activities align with the TRLs that I had discussed earlier. And it's a bit of, there's a range here, right? So TRL 1 through 3 for a couple of the projects, looking at biomarkers of exposure for PFASs and sulfur dioxide, identifying biomarkers for exposure to jet fuel exhaust, and then into establishing a zebrafish toolbox for rapid toxicity screening, and assessment of pulmonary disease using ex vivo and in vivo systems. This particular project is also designed to develop bridging data so that we can move away from some of the more arduous animal studies and work our way into in vitro systems with some confidence on that we are seeing a reflection of intact biological system. Next slide. And so, just as a reminder that toxic exposures, go beyond the burn pits. And I would also like to call attention to the fact that several of these exposures that you can see in this picture, these scenarios, are not necessarily limited to the DoD. These exposures would be extended to first responders, emergency response teams, that come from local, state, and federal agencies. So while we are interested in what's happening to our military members, there is a larger picture of how emergency responders and first responders might also be exposed to sort of similar conditions. Next slide. And so a question might be, how would you evaluate these exposures? And one way would be setting up a health monitoring research program. So this particular program was set up for work with the DoD and the Veterans Administration. And so you'll see some, instances and references to specific militarily relevant compounds. But I believe that the structure of this actually applies to any agency or any group that would be interested in setting up a research program. And, so from collection and sources of data, how do you get it? Where do you get it? Where do you get it from? How often do you retrieve the data? How do you store biological samples and specimens? And we saw some presentations earlier on how they manage those as the BioSend, that data set, and just how tightly they have to manage that, because it's not an endless sample set. It is a finite resource. Next slide. And I think I kind of covered this. So, if you have biospecimen collection, do you need refrigeration? What sort of storage conditions do you need? What are the incentives for the people that you're trying to collect it from? How do you remind them, et cetera? Next slide. Yeah, I know, because we're coming up on time here. And this is just a quick deep dive into the feasibility and scalability of such a research program. And I'm only going to point out a couple things. So under the clinical and epidemiology side, if you're doing that side of the house on the research, you're going to need a core of folks. That's not going to be just one lab doing that work. Under the research side, the toxicants, are they common? Are they rare? We haven't really discussed mixtures. I mean, we're talking cumulative exposures, but understanding the finite mixtures is actually pretty important. And then how would you incorporate, either new methods such as the NAMs, and how would you potentially move away from animal testing for a variety of reasons, one of which is just the speed at which you would collect those data. And also need an administrative core and an information management core to manage all your data. Next slide. Okay, good. This is my last slide other than my thank you slide. So back to the 80 pounds of light stuff, which maybe you all came were interested in. What was I going to actually say about 80 pounds of light stuff? And so the message to that is there's a lot of ideas for chemical sensors and devices which you might be able to deploy. However, you have to remember that there is a person that has to wear that stuff, right? So on the military side, they're carrying a lot of gear with them, and they do not necessarily want to deal with additional gear, or if that gear needs to somehow be managed after its collection period. Is it going to be refrigerated? How is it going to be handled and things like that? And I'm going to also point this back to, this is more than just a DoD issue. If you're going to try to track exposures to first responders and emergency crews, they're also going to have these same problems, because they're going to have a certain amount of materials and equipment that they are carrying. And so whatever those exposure tools, sensors that are being designed, need to be designed with that end user in mind. And I think that's it except for my acknowledgement. There's the link to that full report. And I would like to thank Dr. Matheny and Dr. Williams for their slides. So those slides were put together either from that report or from Dr. Matheny or from Dr. Williams. So thank you.
All right. Thank you very much, Dr. Adams. We are at time, so we'll take questions in the Q&A box. Please enter your questions there, and we will answer them asynchronously. we're on to the final presentation of this workshop. It's from Ms. Robin Taylor. She is the lead for the NIH Common Data Elements repository at the National Library of Medicine. Ms. Taylor has been working there since 2016 as the product owner for the NIH CDE repository. She oversees daily operations, continuous improvement, strategic planning, user engagement, and more. She advocates for the use of CDEs in NIH-funded research and supports NIH users interested in developing, sharing, or using CDEs. Today, she'll be talking about common data elements at NIH.
All right, thank you, Neil. Thank you for the nice introduction. We'll move right through these. I have nothing to disclaim or disclose. As Neil said, today I'm going to be talking about common data elements, or CDEs. I'll talk about CDEs at NIH, including NIH CDE governance and the NIH CDE repository. I'll also talk a little bit about CDEs at NINDS specifically, and leave you with some important considerations for the use of CDEs. So first, we will start with some basics. What are common data elements? NLM defined CDEs this way in a request for information that was issued in 2021. A CDE is a standardized, precisely defined question or variable paired with a set of specific allowable responses, and it's used systematically across different sites, studies, or clinical trials to ensure consistent data collection. In other words, the question is always asked in the same way, and respondents always have the same answers to choose from. And if it's not something if it's a preclinical situation, the variable is always defined in the same way, and the possible responses to that are the same. The most essential thing, what makes the data elements common, is that they're used consistently across different sites or studies or trials. So what's so great about CDEs? Why does NIH recommend them? Use of CDEs makes research data more fair. And if you're not familiar with that acronym, it stands for findable, accessible, interoperable, and reusable. NIH encourages making research data fair. CDEs primarily make research data more interoperable, but at the same time, they also improve data quality, facilitate reproducibility, and make data collection and data coordination processes more efficient. All right, so NIH has long recognized the importance of CDEs and encourages their use in research. They're mentioned in the 2017 Strategic Plan for Data Science, but the COVID-19 pandemic really highlighted the need for coordination across NIH studies to standardize data collection. In the fall of 2020, the NIH Scientific Data Council, or SDC, formed the NIH CDE Governance Committee. This is just the org chart showing where they belong. The governance committee has 8 members from across NIH that were selected by the SDC for this role because of their expertise with CDEs, and includes 2 ex officio members from NLM, of which I am one. All right, here is the charge, the 3-part charge from the SDC to the NIH CDE Governance Committee. The primary charge is to decide whether submitted CDEs merit designation as NIH endorsed CDEs according to criteria that the SDC established, which I'll share with you in a moment. Also, the governance committee works closely with NLM on processes for depositing NIH endorsed CDEs in the NIH CDE repository. And lastly, they review their processes annually. So these are the criteria for NIH endorsement that the SDC handed to the governance committee. So what happens is that NIH groups will submit CDEs that they've developed to the governance committee, who will then review them according to these criteria. And if they meet all these criteria, then they are deemed to be NIH endorsed. So the first 2 criteria here are, in my view, the most important, but also can be the most tricky to review for. What constitutes a clear definition? What types of evidence of reliability and validity are sufficient? The governance committee has been actively grappling with these questions and their practical applications as they've been reviewing submissions over the last 3 years. The third one, human and machine readable format. The governance committee developed a submission form, and at NLM, we have automated the upload and validation of data from that form, which takes care of the machine readable format part. On the other hand, the endorsement review requires a human perspective because it can be very nuanced, especially to address those first 2 criteria that I mentioned. recommended by an NIH body. This is possibly the easiest one to check for. Currently, the governance committee accepts submissions from NIH institutes, centers, offices, research initiatives, other trans NIH groups, but not individuals. Licensing and IP status must be clear, and the submitter must do their due diligence here, researching. At NLM, we do review the documentation provided. And it's important to note, I think at this point, that the SDC specifically said that the governance committee should not adjudicate the scientific merit of the CDEs submitted. The assumption is that the group that is developing them is the subject matter experts, and they will be the best judges of the scientific merit, but the governance committee is using these criteria to just make sure the CDEs are well specified and well defined and meet these standards of data science, if you will. All right, so what does this look like in practice? This process starts with the submitter submitting a collection of CDEs. And like I said, the submitter must be a group from within NIH. The CDEs first go to the governance committee and for sort of a first quick check to see, is it something that they can review? Is it coming from within NIH, for instance? I think almost everything gets accepted at this point because everything that we've received so far has come from NIH and meets all the basic criteria. At that stage, it comes to my team at NLM where we do quality assurance. It says checklist here, but we've moved that process. It's no longer manual. It's now this online data validator that we use to help us, which really speeds things up. And we work with the submitter. We go back and forth here. I reassure everybody that nothing just cruises through without errors. There's always something that we need to discuss. So my team will work with the submitters to address any errors, and that means just formatting errors or missing data, that kind of thing. When the CDEs have passed the QA process, they move over to the governance committee for that review, which is according to those criteria that I just shared. And similarly, the governance committee is going to work with the submitter. I don't think anything has just come through and passed on the first try. There's usually some kind of clarification or slight revision that's needed, but the governance committee will work with the submitter and hopefully get them to resubmit and pass. And when the CDEs are deemed to be endorsed, they are published in the NIH CDE repository. And for each of those reviews, I should mention that it's a panel of no fewer than 3 governance committee members. And yeah, like I said, the governance committee really views itself more like a coach and less like a gatekeeper. They want people to succeed, so they try to work with them. Okay, now I'll talk a bit about the NIH CDE repository, which was launched in 2015 to provide access to CDEs and forms that are recommended by NIH groups. And it's always been hosted and managed by NLM, but it is an NIH-wide resource. Currently, the repository has over 24,000 CDEs. It includes CDEs that are NIH endorsed and CDEs that are not yet endorsed by NIH. In other words, they were added to the repository before the governance committee was established. So NIH endorsed CDEs, as you can see, they make up a very small percentage of all CDEs in the repository right now, but that number is growing. So there's 171 in there, but there's 500 more that have been endorsed and are being prepared for load now. And I think there's 1,000 more that are in the review pipeline process. So we do expect that number to grow soon. And just to explain the 24,000, before 2021, the CDE repository accepted all NIH CDEs that were submitted to it, regardless of format. We had only minimal data requirements. And we do continue to host those older CDEs that have not yet been endorsed. Just because they haven't been endorsed doesn't mean that they're not valid or useful. They just haven't been through that process yet. And so we continue to host those anyway. And then since 2021, we only accept CDEs that have been endorsed by the governance committee. We flag them with a gold ribbon, like you see here on the screen, to distinguish them from CDEs that were submitted prior to governance. And I also want to clear up an important point right now, because sometimes people misunderstand the nature of NIH endorsed CDEs. NIH endorsed CDEs are not required by NIH. At this time, there are no NIH-wide requirements for CDEs. NIH endorsed CDEs are just meant to show that they've been vetted by experts and deemed to be high quality. That said, individual projects may require the use of CDEs by their investigators, and that may or may not include use of NIH endorsed CDEs as well. Okay, this is just a little screenshot what a CDE looks like in the repository. A typical CDE record has a lot of metadata associated with it. This slide shows just some of the basics, and it's an NIH endorsed CDE. You can tell from the gold ribbon there. This was submitted by the NIH-LBI Connects project about oxygen support. Another feature that I just want to highlight quickly is forms. Forms are just simply groups of CDEs. Sometimes CDEs are combined to create a form in the repository. That could be a form that's created for a particular research purpose, or perhaps there's some CDEs that must be used together. Sometimes forms are sort of recreating standardized instruments like the PHQ, for instance. I don't know if you know the PHQ, but that's the example I always give. But other kinds of standardized instruments and assessments, they might look like that, or they might be something that the submitter has just put together for their particular research purposes. So in this case, the one we're looking at, this is a form with simply 2 CDEs in it, but it's 2 that have to be used together because the way this project is collecting age data is it asks for age, which is a number, and then it asks for age units as a separate question, which could either be years or months, depending on the age of the participant. And so they are put together in form. And this is a special kind of form. I don't want to go too in the weeds, but this is a special kind of form called a bundle. And a bundle really, the CDEs in a bundle must be used together. They're not considered valid otherwise. The big difference between the metadata for forms and CDEs is that forms will have copyright status indicated. This CDE is just freely available without restriction. If it were copyrighted, you would see that indicated on the screen there, and there would probably be links out to more information to learn about that. Okay, that was an extremely quick look at the NIH CDE repository. It's all I really have time for today, but I'm going to point you to some CDE resources that you can access on your own time. First is the link up there for the repository. Below that is it's about 1 hour self-paced tutorial just on CDE basics. It has a little bit about the repository, but it's really just about the basics of CDEs to begin with. And then the second item on there is standardize your research data with the NIH CDE repository. That was a live class that we offered 3 times, and you can watch the recording of that class. But I actually recommend trying the on-demand version of this class because it's a version of the live recording, but it incorporates exercises sort of side by side with the repository, so you can try searching and doing other activities like while you're listening to the class as well. And if you need any help, that is the NLM support link right there on the slide. And then I forgot to put it on there. We also have a listserv. I will make sure that I get that link out to you somehow to the group. Maybe I could put it in the Q&A later. It's just a very, it's an infrequent listserv. It's just announcements only from me. So yeah, what that is. Okay, now I promised I would talk about CDEs at NINDS, and I'm not an expert on NINDS CDEs, but I've worked with my colleague Carolina Mendoza-Puccini for quite a while now on CDE governance, and she asked me to present some things on her behalf, so I'll go over the major points that she would ask me to cover. This is another place to look for CDEs. So this is the NINDS CDE website. The URL is here on the screen, and a couple screenshots just show you what it looks like, and then the different kinds of diseases and domains and topics that you will find CDEs on within there. So that is another resource where you can find CDEs. I just want to pause there actually and say I'm presenting these 2 resources today, the NIH CDE repository and the NINDS CDE website, but there are other resources out there, other projects using CDEs. I heard somebody mention FITPR earlier today. FITPR has CDEs. These are not, the things I'm showing you are not the only places to find CDEs. Okay, I'm going to talk a little bit about developing CDEs, and this is not an NINDS specific process on this slide. This is sort of a high-level view of what's involved in developing CDEs. The first step is simply to decide that you want to develop CDEs for a research initiative. The sub-bullets there show some groups that might be involved in that decision, the kinds of groups that you might engage with if you were making that decision. The second step is to choose the CDEs to use. This makes it sound very straightforward and simple. It's usually a lot of work in this process, Groups usually have experts selecting from existing CDEs or developing new ones, and the broader community is engaged, and they have to be all well-defined and well-specified. So that's where the bulk of the work, I would say, is in developing them. And the third step is to approve and require their use. So once the CDEs are approved by the organization, then you want to require their use as well. Many groups recommend CDEs, but I hear that if they're not required, that they are less likely to be used. And there's different ways of doing that. I know some groups will put them usage in like the milestones for a project or something that might be like somewhere in the terms or the conditions of their award that the investigators will have to use CDEs. Okay, so at NINDS, this is Carolina's slide. This is what it looks like at NINDS. So first, the CDEs are identified, as I said, by experts in the community. And then all these other groups are engaged with. So first they engage with other NIH institutes and even other federal agencies, CDC, DOD. They even go beyond that and look at international data standards and see if they can align with those whenever possible. And then they engage with the patient advocacy groups and the industry and others in the community to get their feedback. And then that's all part of, I think, the development process and selection process there. And once those CDEs are established, then NINDS continues to provide guidance, review, oversight, and updates. And I'm not going to read every step here, but just to give you an idea, these are the many steps that happen in that process, the different kinds of meetings that have to happen. And Carolina estimated that the process would take about 18 months. I think it could easily take longer, depending on the scale of the particular project. So it's not speedy, but it's something that needs to be done carefully. Okay, this is the last thing to leave you with. Carolina wanted me to highlight a few considerations for CDE use. So one, there's a learning curve for both investigators and program staff. So learning, I mean, there's from the development to the requirements and the enforcement to the actual usage in the field by investigators. There's something for everybody to learn. If they're the first time through, it might be more difficult. And coordination is important to avoid duplication of effort. I think this speaks to the process of developing CDEs. We have, a lot of silos at NIH. We talk about that a lot, and we find that similar or not similar groups, really disparate groups across NIH will sometimes be developing very similar CDEs, and we want to try to coordinate more to avoid that duplication of effort. That said, sometimes different groups might develop their CDEs in slightly different ways. They might be trying to measure similar things, but they might be measuring it in different ways or capturing that data in a different way. And so sometimes there might be more than one CDE on a certain topic, and there's not one right one necessarily. It's just what's the best one for your particular research question. And Carolina wrote, sometimes it's hard to retrofit existing data to new CDEs. I would say it's almost always very hard to do that. It can be done, but it's difficult. So it is definitely best to identify and use CDEs from the very start of a research initiative, and then your data that you gather is going to be fair and interoperable. And I think that's the very end of my presentation. Thank you very much.
Okay. Thank you very much, Robin. Okay, so we have a question in the chat. I'll encourage other people to also ask questions in the chat. We have a little bit of time. So this one is, data field definition and domain standardization is very valuable service. For example, units of measurements. Thanks. What are the criteria again when bundling CDEs, like age, for example? I'm not sure I fully understand.
Yeah, can you repeat the beginning of that question? I'm not sure I got it either.
Okay. Well, I think the beginning was a comment. Thank you for the work in domain standardization, field definition. And the question is if you can repeat the criteria for bundling CDEs.
Oh yeah, sure. It's not. Yeah, it's really very straightforward. A bundle of CDEs just means multiple CDEs that must be used together in order to be valid. So in the example I gave, one was a number, but there were no units of measure attached to that number. So it's not something that's useful unless you have those units of measure attached. And so there was like there's one CDE for the number, one for the unit of measure, and you have to use those together to make that data meaningful.
Okay, thanks. And then I just have one question. So, you know, as people, if you're developing CDEs, what's the recommendation for kind of trying to look at the existing CDEs, let's say at NIH, or maybe at other data resources and using those? And I guess maybe there's the opposite question, which is what happens when you're using the same name for the CDE, but they're defined differently?
Yeah, so that's an interesting question. We have something called like reuse in our repository where different groups can reuse the same CDE, and there can be slightly different, very slight wording differences, I'll say, but there can't be any substantial differences or it's not considered to be the same. Like the meaning of the CDE must be the same. So if it's defined differently, I would say those may not be the same CDE after all. The definitions really need to be, maybe they don't need to be worded exactly the same, but the meaning must be identical for them to be the same. I feel like I forgot to answer the first part of your question. That was like a two-parter.
Well, I think it's fine. I guess it was like, should people look to see what are existing CDEs before, I guess trying to, submit and validate their own?
Yeah, absolutely. I mean, and just be clear, like individual investigators are not submitting CDEs. This is something that would be developed at a higher level than that, like a branch or a program level, or even an institute level at NIH. So individual people are not submitting their CDEs. CDEs are, I'm not going to go into detail on steps, but I would say you can search the existing resources, like the ones that I mentioned in particular, NINDS, and also at our repository, NIH CDE repository at NLM. I believe that NIEHS might have some useful resources too. They have a database called DR2 that has to do more with, well, it's more disaster-related research, so maybe it's not applicable here, but there are other resources out there. But start with NINDS and search and see what you can find. And the idea is that you would use CDEs for, I think, as much as you could, but then my understanding is that you're allowed to add on additional questions for your own research project, in addition to that. But if there's certain kinds of data that everybody is collecting, then that should be done the same way for consistency.
Okay, thanks very much. And, thanks everyone for wading through my questions. Okay, we'll now move on to the panel discussion. I'll just remind everyone, you can continue to submit questions in the Q&A, and also we are recording this workshop and we will make the recording available. Okay, so now for the panel discussion, we have two panelists joining us in addition to the speakers today, Dr. Daryl Hood and Dr. Caleb Finch. Dr. Daryl Hood is the Professor and Dean's Fellow for Diversity, Equity, and Inclusive Excellence, Environmental Health Sciences at Ohio State University. Dr. Hood has conducted his career with a specific focus on toxicology and its impact on community health. His expertise is in dissecting the molecular mechanisms of environmental pollutants on neurological development and health, and examining the intersection of the built, natural, physical, and social environments. Dr. Caleb Finch is the Arco William F. Kishnick Chair in the Neurobiology of Aging at University of Southern California. Throughout his career, Dr. Finch has used biochemical and genomic approaches to study the physiology of brain aging processes. Dr. Finch's recent work focuses on gene-environment interactions of ApoE alleles in the context of the Neural Exposome. And today's panel discussion will be moderated by Dr. Ekemini Riley, the Managing Director, Founder, and President of the Aligning Science Across Parkinson's, and Founder and President of the Coalition for Aligning Science. She has designed and facilitated several multi-sector think tank sessions to inform the strategic deployment of philanthropic capital, crafted research programs, and seeded multi-funder collaboration. As president of CAS, Dr. Riley sets overall strategy across major philanthropic portfolios focused on accelerating discovery and therapeutic development in biomedicine. I'll turn it over to you, Dr. Riley.
Thanks, Neil. And thank you, Dr. Fitch and Dr. Hood, for joining us today. So before we get started with any big questions, I wanted to give you both the space to react to any of the presentations that we heard today, lots of resources that we heard discussed. So, given both of your backgrounds and what your work is focused on, Daryl, I'll start with you first, and then Caleb, I'll hop over to you to just get your general reactions before we jump into questions.
Wow, thank you very much. Oh, go ahead, Cleve. Cleve, you can go first. Go ahead.
Well, I'm delighted to be on a panel with Daryl, whose work on benzapyrene I've followed, and multi-generational effects that are scary.
Yes, I agree.
Well, I'm struck by something that Tim Buckley said, and there's too many chemicals to measure. We can't measure our way out. And this recalls discussions over the decades with, well, what is the carcinogen in tobacco smoke? And of course, the conclusion was that there was a multiplicity and not a single one would account for it. And there was also interactions with the particulates that are a separate physical and chemical set of questions. And I don't have high, with the biochemist background, I don't have high expectations that we'll be able to resolve any specific chemical with the multiplicity of effects, because there are so many interactions and synergies. Just one example of that comes to mind from John Sammet's work when he was at USC, who got me interested in air pollution. He found, examining the role of cigarette smoke in lung cancer, that air pollution also had a dose-dependent interaction with cigarette year smoke measured, and that the interaction was supra-additive by about twofold, separately from each considered by itself. So I anticipate that there will be a lot of importance in looking at synergies of these the various things that were discussed today, and that's an immense agenda for which we need new generations of animal models. And come back to the research cigarette that was so important in early animal studies in the 1970s, 80s, and 90s. And there's now at least 10 different animal model exposures to particulate air pollution components. And I think and then there's the third component that was mentioned of environmental noise and the social context of these exposures with really important issues about green space. So I think we need to build some new animal models that goes beyond the accessibility of most individual labs standing alone. So those are some of my thoughts.
Thanks, Caleb. And on to you, Daryl. Daryl, before we start, I just want to mention to the rest of the folks attending that they can go ahead and start putting questions into the chat, and we'll keep the conversation lively and going. Sorry about that, Daryl. Over to you.
No, that's okay. Thank you very much. I'm certainly in agreement there with Khalid, certainly. More importantly, or just as important as those animal models are, I think that Tim hit the nail on the head, certainly when he delved into the area of the cumulative nature of all of these interactions between chemical stressors and non-chemical stressors.
Mhm.
And so for me, and David started out the day so well in terms of contextualizing for us the challenge that lies ahead. He put it right out there. I mean, and the talks today demonstrate that the possibilities, the questions, but also the challenges. And I'm sure you'll get to that very, very shortly. For me, the CDE section that we just heard, common data elements, seems to be front and center in terms of low-hanging fruit. And I was so disappointed, but yet happy for Pat Brennan when she retired recently from the National Library of Medicine. And going back to David, you know, we're following exposomics, seems to be following the general trend with respect to genetics. And so this is presumably the next biggest thing on the horizon. Being on council at NIEHS gives me a front row view to all of the innovations that NIEHS is putting forth. And I'm happy to see NINDS and their efforts as well, as David said. But we've got to get to a point where these common data elements we have to agree on them. We're still trying to decide on a lexicon, for an example, a common lexicon. And so the future is bright, but we have some challenges, and I'll let you move to that particular section of the discussion.
Lovely. So what I'm hearing are, one, the synergies across the various aspects discussed today, the cumulative nature of both chemical and non-chemical exposures, as well as the social context of all of these exposures, the sort of main themes that are being drawn out from the day. Which is important because the first question I really have for you both to react to, and then I'll open it up to the broader panelist group that we have on the call here today, is what you all think are the must-have tools and resources for exposomic research, really specifically to draw out those key elements, right? You're talking about synergies, cumulative nature, and social context. Getting all three of those things, you know, within the tools and resources to study the exposome seems like a massive challenge just from listening to it from the outside. So want to hear from both of you as experts. What do you think are the most important tools for this?
Errol?
Thank you very much, Caleb. Well, I think that certainly it's very, very complicated. We know that. And so with regard to understanding the interaction of chemical and non-chemical stresses from birth, from conception to death, right? Or, you know, cradle to the grave, from the womb to the tomb, you know, there are all types of contextualizations out there right now, but that's daunting. It really is. Right. And so we've got to start simple. And I think that, first of all, we as a community in the academy and all around need to realize that, you know, when you're talking about the exposome, it's really, as Christopher Wiles so aptly indicated, he's the father of all of this, in my opinion. We've got to think about the endoexposome versus in consideration for the ectoexposome, right? Yeah, and so that is daunting in and of itself. Then you have to you have to be able to contextualize a framework that is going to be able to handle both. And that is where I've been working for the last 10 years with my colleagues, Paul Juarez, and the community of practice at Meharry Medical College and Vanderbilt University. And so that interaction and partitioning, and then the framework that all of that goes into, I think the public health exposome framework is aptly suited to handle that, the ectoexposome as well as the endoexposome. So you're talking molecular level pathways, biomarkers, and the like. Tim talked about that a lot. And then, of course, that social context part,
Right.
Madam Chair, that you're speaking of. Okay, those two actually interact with each other. Okay, and so a social ecological life course framework that's undergirded by, you know, exposomics, of course. We're talking endo there with respect to the molecular level adverse outcome pathways, I think is something that should stand front and center in that regard.
Well, Caleb, you mentioned new animal models. If you could expand on the types of animal models you mean, but then also, you know, broader, what other tools and resources you think are must-have?
Well, most of the experimental work has been done, I would guess 90% of the publications on rodent exposure.
Mhm.
The literature is not very large. I mean, maybe 500 or 700 experimental studies have been published in one way or the other. And then these also include some in vitro cell in a dish studies, which have some level of informativeness, but of course they don't represent the reality that inhaled particles, which is just part of the exposome, most of them get trapped in the lungs, or the gases such as ozone, that really doesn't, by any data I've seen, reach the deeper alveoli because it gets trapped in the mucosa along the way. So there's various dimensions of this that I think need to be pulled together in a multi-institute meeting. I tried to organize one and it didn't get funding because I don't think they really understood what Matt Campton and I were trying to do. To identify what other animal models are useful. There's a parade of transgenic mice, and then there's some new transgenic rats with human genes that are proving very useful. I'm reluctant, but I must say that we need also to consider primates. And besides the rhesus monkey, there are smaller primates, the dwarf lemur, that merit some consideration. They have a lifespan of about 10 to 12 years. And there may be a group of studies that should be engaged in that. But I think that there's a need for a broader discussion, multi-institute, to review existing models and the ones that might be considered. Now, there's an aspect that animal models don't obviously allow, and that is something that I don't believe has been mentioned, that the socioeconomic gradient on air pollution exposure and cigarette exposure and many other adversities is associated with a 15-year difference top to bottom in lifespan and in the prevalence of Alzheimer's disease. People with less than high school education living in poverty have a 12 to 15-year earlier prevalence of dementia, and that's been documented in at least 3 different populations, and it seems to be on a linear scale, which is equally puzzling. I don't know what SES, environmental, external, and internal, really follow an education, human and household wealth gradient in a linear fashion. But that's what the data looks like.
That's right.
So those are some questions that in discussing animal models, I think we need to calibrate in the beginning that there are some questions of equal importance to human populations that will be much less accessible in the pragmatics of doing animal studies in labs. And then there's another point, we can talk about this later, and that is across the SES gradient in air pollution and adversity in the exposome, there must be gene environment interactions on a very large scale. We have a little hint of that with some of our work on transgenic mice with ApoE4 that are more vulnerable to air pollution in our model. And there are some population studies that look at that too. But it's a very early topic, but it's obvious considering the epigenetics that were mentioned in one of David's early slides. I don't believe anybody's mentioned much about epigenetics. There's a major set of questions that have to do with gene environment interaction. And of course, there's a great deal of attention now to DNA methylation, but I'd point out that another epigenetic component is histone methylation and acetylation, which is rarely, if ever, considered in the same context of the DNA methylation in those same cells. And tell me otherwise, but I don't believe we have as convenient ways of measuring histone and acetylation and methylation. But that certainly is part of the genomic transcriptional package that has environment and interactions as well as the DNA methylation. That's enough for a moment.
Absolutely. So I want to open it up to our other participants on the call today. Please feel free to come on camera and provide some of your own short remarks on what you feel are the must-have tools and resources for studying the exposome. And I also do want to make sure if someone has a response to Chris's question around leveraging organoid models for exposome research, that I would love to sort of hear that brought out as well, because that could possibly address, Caleb, your question on the epigenetics and thinking about histone acetylation, methylation, and et cetera. So is there any one of the participants? So Debbie, and then I believe we have Yushia, who also has a comment. So Debbie, go right ahead.
Yeah, hi. What I'm delighted to hear through this today is sort of what I think is moving beyond exposomics, because it isn't just what's out there, it's how we put it together and study it in a cumulative framework. So to me, it's always been about cumulative risk, not just what you're exposed to. And I think one of the things, it clearly is a hard problem, but I'd like to put out there the fact that there are some algorithms that have been thought about in this context. And I'm going to go back to one that came out of a National Academy of Sciences review or book back in, I can't remember at this point, it was on phthalates. And the National Academy asked us if we should be studying phthalates cumulatively. And we said, of course, that's really silly, but let's talk about cumulative risk. And I'll remind people that we have huge databases already in which we've studied the effects of individual chemicals and individual stressors on disease phenotypes. So for example, let's say I was worried about cumulative risk for hypertension. I can already go to that literature and identify lots of different risk factors that have been tied to hypertension. Those are an obvious mixture study right there. And I can do that kind of mixture and then say, which of these are producing the greatest magnitude of effects? Because now that takes me to the policy side of the equation. So thinking about it in a backwards sort of way, from disease back to what risk factors have been identified and how can we study those in a cumulative model, is already an algorithm to think about. With respect to organoids and in vitro approaches, there are a couple of issues there. Number one, I don't know having done work on stress and chemical exposures, how you would develop a in vitro approach for what, in the economic literature is called resource deprivation, poverty. I don't know how you would do that, frankly. So the other thing to think about is when we're talking about risk assessment, as it's done by EPA, they basically identify the sort of most sensitive endpoint among different endpoints to begin with. And again, unless you're running 1 million NAMs, I don't know how you would get to that without the in vivo data. And I don't know how you put all these contexts into a NAMs approach. So just some thoughts.
Lovely, thanks, Debbie.
Thank
I
you.
think it's also worth considering that perhaps there may be some things that are not modelable, for lack of a better term, in vitro, but things that are more suited for an in vivo environment. So Yushia, you please provide your comments, and then Dave, I know your hand is up as well.
Okay, thank you. I agree with all the previous discussions. And I'm going to say something really biased because I came from the exposure science exposome program. I would say, for exposome research, really, there are lots of must-haves to put things together. And I would like to emphasize the technology platforms for collecting exposure information, exposure data. So by that, I don't only mean chemicals, but also social determinants of health, lifestyle factors, people's behavior, which will have lots of impacts on how the person will be exposed to and what they're exposed to. Debbie mentioned the cumulative impacts. We need to know, the historical. We don't have lots of ways to know the historical exposure to capture that kind of data. I've seen challenges in all these areas. For example, an epi study tried to capture the chemicals the population are exposed to. They need to do multiple assays, and that burn out the funding they have. So essentially, it's cost prohibitive. So we really need to kind of think about the genomics. You need that kind of platform that can measure lots of chemicals at one time. So that way, you need to get the information, knowing that can feed to animal models about, okay, in the future study, do we need to measure all or some of them, through in vitro studies can help us narrow down the things we can measure. So I think there is a kind of positive, kind of feed to each other, feed the information, between animal study and human study.
Lovely.
And for the historical exposures, I've seen, people try to develop instruments or surveys, try to get the information. But, in the 45 minutes interview, you can only ask so many questions.
Right.
There's a burden to the participants. You don't want them to run away from you because it takes too much time from them. So really kind of innovative approaches to get that kind of information. Because, we can get a neighborhood level information, but you don't know the behavior. Even in the same neighborhood, the exposure, the impacts to individuals are different depending on other factors. So I think, the tools is really important, but it's one of the must-haves.
Right.
To think about the genomics, originally the concept was brought up is because we are lacking of tools, understanding what we are exposed to, compared to, what happens in the genomics world.
Right.
This is not the only one, but I think it's quite important. And as NIEHS, we cover lots of diseases, but our, great interest is on the exposome, the entire environment.
Right.
My thoughts.
Great. So I'm hearing a double down on the cumulative framework, multiplexing of various assays. Dave, we'll hear your perspectives, and then we'll move into the barriers and challenges and bring it back to Daryl and Caleb.
Well, maybe this is related to that as well. I didn't really want to ask questions, but there's one that just keeps me up at night. And when I was talking about getting this together at NINDS, this is going to be so hard. I mean, the bottom line is, and this is a challenge question, are we ready? Are we ready for this? I'm going to steal a word from my old friend Daryl and say, it is absolutely, in my mind, daunting how much information we are going to be trying to put together. I mean, first, just getting them one by one, and then taking those and bringing them together. It's daunting. And so the question was, you know, do we have the tools? That's why I wanted to do this. Do we have the tools? And this is, again, just a challenging. I mean, things like, I don't think we've talked about AI. And AI can be good, AI can be bad. So things like that, I just wanted, as we're talking about this, to keep that in mind. And that's all I wanted to say. I'd rather listen than talk.
Right. Well, I mean, we'll go right off that jump point. Daryl and Caleb, coming back to you both on your thoughts for the challenges of getting work going in this space. And I mean, I will just follow on from what Dave mentioned. You know, with AI, with what I'm learning in this space is that AI is only as good as the data that you have and that you put into any model, right? So, I mean, knowing that and knowing the potential power that we could leverage with AI and machine learning, it's almost incumbent on us that we get the data right. So just, I guess, let's keep the spirit of the conversation high while also understanding and acknowledging what the barriers and challenges are, because as Dave said, they can be daunting. But let's sort of go through some of those challenges. And then I'd like to really challenge a lot of our panelists and folks who are listening in. Drop your comments. I mean, let's talk about how we can start to address some of those challenges to move forward and really tackle Dave's question of, are we ready yet? So Daryl, Caleb, over to you first.
Okay, I have one slide here. Can everyone see that?
Yes.
Yes.
Oh, lovely. It's like you knew I was going to ask this question.
Hey, might have had a
Daryl, if
little
you could
hint.
just swap the displays, go to the button at the top.
Oh, yes, I'm sorry.
Left. Yeah, go right in. Yeah, there you go.
Sorry about that. Yes, and that's not the one. Yes, this is the one. and following up on Deborah's question, my good friend from Rochester, you know, as far as poverty goes, you know, Deborah taught me environmental enrichment. I read her literature, the enrichment. She helped us get going. She took my students up there with her during the summertime. And, you know, once we got a hold of the baseline with respect to what enrichment really is, then that is how I look at poverty in the cage, okay, in the behavioral cage, yes. So there is a way to do that. But here, and thank you, Debbie, for being instrumental in the group in that regard. So yes, we have all this environmental data. We have all this domain knowledge now. It's overwhelming, as Dr. Jett just indicated. The combinatorial algorithms are coming at us from left, right, north, and south. We have these supercomputer platforms. So presumably that's going to help us understand these disparities within the context of the overall exposome, right? Mm-hmm. Okay, so even still, the biggest challenge is what I have to the right here. So we put this is pretty new, came out in January. I think it says December, but actually showed up in EHP in the January edition, I believe. And the tool chain to the right is what the public health exposome community of practice is pushing for all of this data, right? You got to clean it, dirt, right? Garbage in, garbage out, right? So you've got to clean it, you know, organize it. There are feature selection considerations, right? And then, of course, you get to the combinatorial, the dense subgraphs, right? It's my favorite, which sort of reveals some latent factors to you. And then you got to interpret that, do the validation studies, and then comes the input to public health practice and policy. And so until we agree, and this is why I miss Virginia Brennan, and I think the director of the NIEHS is working diligently to get us all on the same page. Gary Miller and his group, Bob Wright at Mount Sinai. I can go on and on and on. Out there at USC, Irvine. I mean, we have people everywhere working on this, but we've got to come to some sort of agreement so that our common data elements are aligned in such a manner that will get us on the same page so that we can then get to some of these other issues. But until we get that right, and we're working, very, very diligently on it, it'll remain a challenge. Caleb. I'll stop sharing as well.
Caleb?
I don't really have anything to add. That was elegant and clear, and it ain't simple, as we all say. The question does occur is, how are we going to train the next generation of graduate students and postdocs to think about this? And each of our labs is in graduate programs that have quite a wide variety of topics and technologies. Is there such a thing as a summer course on the exposome that could be considered? There's summer neuroscience courses that were extremely important in at least my generation of understanding what goes on up here.
Right.
And there might very well be such a platform, multi-institute and foundation approach.
Right. Thanks, Caleb, for lobbing out one of the ideas that we can use to tackle this challenge. So I invite some of the other panelists to offer your thoughts on what some of the challenges are, some ways that we can address those challenges. Valerie, I see your hand up. You can come off camera and come on camera and share your thoughts. Gina, if you want to share your thoughts as well, please feel free.
Yeah, thanks. So I was just jotting down some notes, and we were talking about cumulative exposures. But in the real world, with the exception of some factors, say smoking, that would be a daily exposure for decades, depending on the smoker. For a lot of people, it's a sporadic or intermittent exposure to moderate to high levels of a compound, followed by periods of time where there is no exposure. So I'm not sure how it's like spatially explicit, right? I'm not sure how you address that. And in some certain circumstances, is it worse to have periodic pulses of exposure versus one low level of chronic exposure? And then when we think about interlaboratory experimentation and how one lab may do a set of tests compared to another lab, study validity and the experimental design to include reference chemicals in those data sets so that everybody has an understanding of like their baseline performance in those assays is also pretty important and probably underappreciated. Over.
Lovely. Thanks, Valerie. Gina? Oh, Gina, you're still on mute.
First of all, I just wanted to say how amazing this whole day has been, and thanks all to the organizers for doing it, and you, Ekameni, for running this particular panel discussion. I think it's just so valuable. And to say to David Jett, it is extremely daunting, as you know, but you also know that it's extremely worth it, because once we get these things in motion, the rapidity of advancement of human health will be unprecedented. It will be just amazing, the questions we'll be able to answer that currently can only be answered in small cohorts and samples. limited sample. So I'm really excited about this. I'm really excited. So the major challenges that I see, are things that other people have identified as well, which are, getting the data in the first place, having enough public trust to give us all of this data that we want. I mean, we will need deep and multiplex data sets where people will have to answer questions about where they've been living, what they've been exposed to, and do it in as often and as detailed a fashion as possible. And we really have to earn the public's trust with that data, showing them how we'll treat it, how we'll ask them for permission to use it whenever we want to use it in different ways, et cetera. Data privacy is important. And then the other one is that the culture of science, as a scientist myself, I know that, the kind of the lone cowboy model is what we've gone with. We write R01s, which are individual investigator initiated grants, and then we gather the data and we publish it. And in the past, we haven't even made those data available to the public because there was just no way to disseminate it, and nobody could know what to do with it. And it was a huge pain. So once we build these structures, these infrastructures for handling the data, we'll need to change the culture of scientists to actually use it and incentivize scientists to use it by perhaps, saying, you don't get your next grant until you've uploaded all your data into these databases. But anyway, those are the things that I was thinking of.
Thanks, Gina. Is there anyone else? Any other participants? Chris, I see you've come on camera.
Yeah, I also just saw that somebody had mentioned in the chat, we were just talking momentarily ago about different training programs, and Dr. Gary Miller at Columbia University has put together this really excellent exposome boot camp, he's calling it. It's been going on for a few years, but there's another one coming for 2 days in July. So I put that in the Q&A if folks are interested. And Gary has been really great, and I believe he's also participating on the strategic planning for this initiative as well as should be. So really, really great to have his insight, and can't recommend that enough. I just want to echo everybody else's statements about the importance of putting all of this data together. I think it's incredibly important. We have a lot of really rich preclinical data, but also a number of clinical data and associated existing biospecimens that we showed you today. Just not only that NINDS and other NIH repositories are supporting, but a number of other non-NIH repositories. So there's a wealth of extant biospecimens, antemortem and postmortem data, and a really rich opportunities to really dig in and produce some molecular profiling. But at the end of the day, where does all of that data go, and how can we democratize that, standardize it, curate, and make sure that it's available to the community in a secure but also standardized fashion, so that the entire research community can do secondary analysis and be able to provide additional rich hypothesis generating questions and answers to it. So I'm really looking forward to continuing these conversations and collaborations as we think through next steps.
Lovely, thanks.
Question for David, if I may.
Yes, of course.
To what extent are there interactions of the NIH programs with European research groups? There are certainly groups in a number of the European countries that are university-based or federally based that we all refer to. I'm not aware of the strength of the conduits between these, and the NIH could have a leadership role in this if it doesn't already.
I mean, I was, as I mentioned earlier, that they're really doing a lot in Europe. And we went, and somebody mentioned this thing where we went and all of us went and came up with an idea of how to define the exposome, and Europe was there, at least half of it, maybe not half, but so they are definitely involved. But I would love to turn this over to if hopefully she sees she's still here, but there she is. She has her hand up, and she can give you a much better idea of what we're doing
And
with
Asia and Africa, of course.
Mm-hmm.
Yeah, but you she'll tell you about what we're doing.
Sure. Thank you so much for that question. I mentioned in my presentation, but it was so quick, try to finish on time. I mean, as David said, there are interactions at conferences, workshops. NIHS actually organized quite a few workshops to invite individual researchers from different countries, continents. David was part of those interactions. David was there too. But you're right, kind of as an NIH group or kind of US network, we don't have it currently, but we're going to have one soon. So we just, 6 ICs and offices is supporting this new initiative called Center for Exposome Research Coordination. Essentially, we're going to fund a group to network within the US and also interact with the European program. They have a European Human Exposome Network. And Japan, there's the Japan Children's Health Study called JAX. I might not be saying exactly the name, but there are some international efforts. Has been interacting in the past kind of informally, but moving forward, we'll purposely organize meetings, organize the community to build a consensus on the framework, on the resources and the tools that can be used now to support exposome research and make that widely available to the community. So it's going to be a 5-year program supported by NINDS, NIHS, NIA, and also NINDS, and Office of Women's Health Research, and there's NCI as well. So that's coming soon. Thank you.
Very soon. And you say, thank you very much. I can't wait. And is it not true that potentially you indicated, NIEHS is just going to fund one, right? One center, one coordinating center. Okay. So, we need to keep this going. Yeah. Even though Dave, thank you for being a partner there, but let's keep
thinking about the Africa and I don't know a lot of it about that, but I think there is some interest in Africa and in not the other one. Oh, and Japan, absolutely. I'm going to give the talk in 3 weeks down there. So they're doing some stuff as well. So it's really spreading around. It really is.
Yes.
Well, great. I think we have about 3 more minutes left in this discussion. So I want to once again leave it open as if anyone on the panel has final thoughts. Caleb, Daryl, if either of you have final thoughts before I go back through and summarize some of the big points that I've heard in our conversation and then turn it back over to Neil and Dave. Closing thoughts, Daryl, Caleb?
Yeah, that's Daryl. I think that just let me make this okay. Yes. Is it showing properly this time?
Yes.
Okay, yeah. That was a huge point, Caleb, that you brought up earlier. And from our vantage point, we are very, very concerned. We have some papers coming out soon on AI, and this is just one of the culminating figures from that paper. Nice. Where the emphasis that our group will be sort of focusing on going forward is the EHR. Regent Street, the Indiana Data Bank, all of the federally qualified health centers in America under one roof. This is good. And so here is sort of that cycle that if we don't sort of look at these guardrails and some form of keeping the train on the tracks, then we're going to be in a bad place. And so there's already underrepresentation with respect to minority input into these large language learning models. So we're also trying to figure that out as we move forward. And this is just some of the disproportionality that all of that leads to if this is not addressed in the short term, particularly in this age that we're in. So I'll stop there. Thank you.
Thanks, Daryl. Caleb? Yeah, I mean, truly, that hand clap was right on time. That was a fantastic figure, Daryl. Caleb, do you have any closing remarks?
Well, just reflecting on the lifelong concept, and I would point out that the egg we came from was in our mother's ovary before she was born. And that means, to some extent, we are potentially influenced by our grandmaternal environment. And that Debbie Koresh-Lechta's work on lead has shown 3-generation effects. And, I think there's potential here to expand the time domain in a conceptually important way.
Lovely. All fascinating thoughts. And thank you both, Daryl and Caleb, for being our esteemed panelists for the afternoon. So just quickly to summarize, we've heard a number of different points, but I think some of the ones that jump out at me are really thinking about the cumulative nature of chemical and non-chemical exposures and really expanding that framework of the exposome to not just the things that we've been exposed to, but how we handle it over space and time. The other pieces we've heard are the need for new animal models to capture animal and cellular models to really drill down into gene environment interactions. The next piece here is on what Yushia mentioned, the multiplexing and scalable nature of models and databases that we need to make sure we are bringing in. And then finally, this concept of the challenge of the acute, sporadic, intermittent exposures that one can experience versus chronic exposures, and just thinking about how we capture all of that from databases that we're all mining, from models that we're using to investigate these effects. So, I mean, it's a large topic, Dave, as you mentioned, daunting, but I think amongst the group of really intelligent folks that you have brought together at NINDS with this subgroup, I think that we're in a place to really tackle it. So thank you.
Thank you.
Thank you, Madam Chair. Yes.
Over to you, Dave. Neil. Dave, you're on mute.
I just have been thrilled about how much we have we've made progress. We've made serious progress with both of these two things we've had. One was the first on team science, and this one has been so helpful for us to really get this going. And the other thing is the collaboration that we're doing with our friends at NIH has been just absolutely fantastic. And I'm hoping that we can get this really going now and really start to make a difference. So thanks everybody for all your talks, and thanks for all of our people that listened to us. Neil, if you had something to say, please go for it. But thank you, everybody.
Well, I think that does it. Thank you, Dave.
All right.
Take care.
We'll see you.